thank you to the organizers to invite me to this conference and giving me the opportunity to talk to you about the globalization of green chemistry within the pharmaceutical industry. I'll try to show you some advancements as well as some opportunities here. Let me start, and first of all, can you hear me okay? Okay. So first of all, let me start with a question. So what is a sustainable future? I'll try to give you an answer more from the pharmaceutical industry, but I think it's true for related industries, and it's true for the academic world as well. The pharmaceutical industry will be expected to meet affordably the needs of patients around the world at a cost they can afford, while we are minimizing our environmental footprint. Well, what do we have to do in order to achieve this? We have to balance, and balance more as we're currently doing, social, environmental, and economic needs globally, and most importantly, across generations. If we look at our future world, well, within the next 20 years, there will an increase in population to about 9 billion. That's good news. We have more consumer of our products. Well, the bad news is, if we continue the rate of consumption of our energy, we will need 2.3 Earth. Well, guess what? We are not getting additional Earth. So we have something to do about this. And I think this demand really needs transformational change, transformational innovation, how we use our energy, how we develop greener processes, also how we travel. So what can we do about it? Since this is a conference about green chemistry, let's start with green chemistry. So what is green chemistry? There are numerous definitions, and I'm just showing you one of those. It is the design of chemical products and processes that reduce or eliminate the use and generation of hazardous substances and waste. It creates inherently safer processes and increases process efficiency, while overall it's minimizing the environmental footprint and impact. I hope you all are familiar with the 10 or 12 principles of green chemistry and green engineering. I have illustrated them here in this graph, and it's all about using catalytic transformations instead of stoichiometric, avoid waste, be environmentally friendly, be energy efficient, safe, look for sustainable ways of doing your processes, being atom efficient, and use non-toxic chemicals. Within Merck, and I know that many others think that good science is really the key to sustainability, green chemistry, and low-cost manufacturing across the globe. David Constable at one time has said, and David Constable is the current director of the ACS Green Chemistry Institute and has decades of experience with sustainability and green chemistry, that he, once a process is greener, is not conscious of anything more costly. So if we focus on developing the best chemistry, then almost always this leads to the lowest cost and the greenest process. How do we get there? Well, I think there are 3 key elements to achieve this. This is education, global collaboration, and innovation. We have to change the way we are doing our processes. We have to think out of the box, and I hope I can show you a couple of examples which hopefully will inspire you all to do the same. So let's go to education. A couple weeks ago, I came across the University of Massachusetts Boston Green Chemistry newsletter. The first issue, as you can see, and they were announcing that the green chemistry track in the chemistry PhD program at the University of Boston was established in 2002, and it is the first such program in the world. Students obtaining a degree from this program will be prepared for conventional chemistry, jobs in industry, government, and academia. And they will have the knowledge to drive this innovation, what we really need, thinking out of the box. I think a lot has been done in the academic world to promote green chemistry, but we are still far away of having green chemistry courses in the curriculum of engineers or chemists. And I am often part of hiring at Merck, and one of the part is we have them give a presentation, and I still see that they do present chemistry, great chemistry, but they are using benzene, chloroform, CCl4. I think, and it's all up to you as well, to change this. I think we really have to think of our solvents, because you will see that solvents are really contributing a lot to the waste we are generating. What else can we do? Well, we can look for organizations which are driving green chemistry. And I'm showing you a couple of them. It's not a complete list. I'm showing you the ones I'm most familiar with. So one is the ACS Green Chemistry Institute Pharmaceutical Roundtable. You are seeing here the current membership as of August 2014. It was, as you can see, it's about 15 pharmaceutical companies, but not only pharmaceutical companies, also suppliers. And as Codexis, Cubis, Dr. Reddy's, or DSM, and you can see these pharmaceutical companies don't do only small molecules, but they also do biologics like Amgen or Merck or Pfizer. In the box at the bottom of the slide, you see the 3 companies which have founded or helped together with the ACS Green Chemistry Institute to get this organization up and running. It was founded in 2005, and next year we will have our 10th anniversary and a lot of special events coming up to celebrate this 10th anniversary. So have a look and see, and maybe you can attend one of these conferences sponsored by the ACS Green Chemistry Institute Pharmaceutical Roundtable. The Pharmaceutical Roundtable has 4 strategic priorities. One is collaborating globally, because all these pharmaceutical companies and suppliers are global companies. Collaborating globally always has been a strategic priority. All activities of the roundtable are global with respect to participation and influence. We have 1 meeting per year outside of the US, normally in the spring, and we do sponsor research lectures at company sites in the EU as well as in the US. The second strategic priority is informing and influencing the research agenda. The way we do this is we give awards of research grants, and we partner in those with NSF. What the roundtable does, it publishes the key green chemistry and engineering research areas they want to have an impact in and influence on, and then we are calling for people who are willing to do research in these areas, and we award grants. And normally we do this in the autumn, so you should see, timing-wise, you should see pretty soon calls for submissions for these award of research grants sponsored by the Pharmaceutical Roundtable. As I said, education is very important, so the Roundtable sponsors workshops for undergraduates, graduates, and teachers, and doing this in the US as well as in Europe. And we are co-authoring green chemistry articles of interest to the pharmaceutical industry. So a subgroup is getting together, screening on a biannual basis, screening the literature for more greener processes, and then putting it all together into one article, and then this article is published in OPRD. Hopefully you all already have seen them. If not, look out for them. They really give a very nice perspective. Lastly but not least, the roundtable is developing tools, giving you an instrument to really be able to do green chemistry. As I said, solvents are really important, so we do have a solvent selection guide. This is accessible at the link I have here, free of charge. We're also working on a reagents guide. This is greener conditions for commonly employed transformations, for example, amide formations. And we have a calculator for process mass intensity, PMI. I'll come in a slide later. Will explain what PMI is all about, but it's a measure to see how green your process is and how you are doing. Again, this is accessible. You can use it free of charge at the link I have here on my slide. There are some additional external collaboration opportunities. Again, this is not a complete list. There is an IQ consortium. This is a consortium which is striving for innovation and quality in the pharmaceutical industry. We do have a green chemistry working group in there, and this green chemistry working group seeks engagement with the FDA, so also look for opportunities where what regulatory can do to enable green chemistry and green engineering. And we also share best practices. We are giving webinars, for example, showing important elements which is needed to have an effective green chemistry and green engineering program in your company, and you also can take these thoughts for academics as well. There is the Innovative Medicine Initiative, which is mostly a European initiative. One part is Chem21, where there are 6.4 million for improved catalytic euros for improved catalytic and flow chemistry method, novel biocatalysis, synthetic biological approaches to enable useful cascade reactions in cells, and improvement in education. And this is done from the pharmaceutical industry as well in collaboration with universities, mostly in Europe. And you're all probably familiar with the National Science Foundation. There are grant opportunities for academic liaison with industry. I'm just listing one example because Merck is involved here, from Professor Maleska at the Michigan State University. It's all about CH bond activation and functionalization methods for medicinal and process chemistry. There are many more other organizations, and I think they are a great opportunity to collaborate globally and to really make an impact and change the rate of consumption of the energy we are currently showing. When do we apply green chemistry? Well, the idea is to integrate green chemistry throughout the life cycle of drug research and development and beyond. As we have heard, green chemistry is the best chemistry. The best chemistry is not only cool chemistry, that means, nice synthetic ways, innovative approaches, but it's also chemistry which tries to decrease the environmental footprint, and because only if you look at all aspects, you will have the best chemistry you can think of. So early awareness can lead to large paybacks later. I don't know how familiar you are with respect in the pharmaceutical industry, how these new drugs are developed, but as you can think, it starts very early on in discovery, and then it goes through phases where it goes into the clinic, and then with an increasing population, then the end point is you launch your program, your project or your compound, and then this is about phase 3, and then phase 4 is after the product is on the market, and this is then not done, but you still look at the synthesis, you look at possibilities to make it greener and to reduce the environmental footprint. How do we calculate green? I already have mentioned this, so if you want to know where you are and how well you are doing with respect to green chemistry, you need a measure. There are many measures out there. One is the Lifecycle Assessment, which not only looks at raw materials, at your input and at the quantity which comes out, but also looks at other areas like packaging, like the whole life cycle of raw materials. What we are doing at Merck currently is we use an easier metrics, and this is the process mass intensity. This is the quantity of raw material you put in and divided by the quantity of API. So if you have a PMI of 100, that means that 100 kilogram go into as raw materials, as solvent, as whatever, as waste in the end, and the 100 kilograms make just 1 kilogram of active ingredient, what is in your drug. So it includes all steps in the synthetic pathway from commonly available raw materials. Raw materials are all materials, including solvents and water, used in synthesizing, isolating, and purifying the API. Within Merck, the typical, what we see very early on from a discovery route, the PMI is more than 1,000. Once we are getting ready to launch the new drug, we want to have our commercial processes less than 100. This is the target. As you can see, this is the graph below, shows you a very typical composition of waste. And as you can see, this is about 56 to 58% of solvents. So this is quite a lot. And then water is about 30%. So if you are taking care and really thinking through about your solvents, what solvents you are using, and also make sure about the water, what you do with the water, you are addressing about 80% of the waste you are producing. Therefore, it's so important that you think about your solvents. And as said, the ACS Green Chemistry Roundtable does have a solvent selection guide, which is free of charge. Take a look there. Really smart choices with solvents are so important later on to be and to drive greener processes. I'm going to show you two examples from Merck. One example is just doing the best chemistry, or try to do the best chemistry. The other one is more thinking out of the box, really looking for innovation, looking for some stuff which you think is not possible, but it is possible in the end. So let's go to Valtegravir, where we had a significant increase in process efficiency in the second generation process. That means second generation processes, you have made your first supplies, you are out of phase 1, the drug has shown that it works, you are currently showing that it's efficacious, and you are going to phase 3 and you are getting ready for the commercial launch. So Isentress is a prescription human immunodeficiency virus medicine. It's used with other HIV medicines to treat adults with an infection. And as we all know, HIV is the virus that causes acquired immune deficiency syndrome, or also known as AIDS. So what we did here, this is the original chemistry. You do see the number of steps on the y-axis, on the x-axis, sorry, and the cumulative PMI on the y-axis. And it's also published in Organic Process Research and Development, ORPD, so you can read a little bit more about that as well. What you can see here is, if you look at the steps, that the first steps apparently are not contributing a lot to the overall PMI, but if you look at step 5 or step 7, this really is contributing a lot. So in order to further streamline the process and make it greener, you really have to look what is happening in step 5 and 7, and this is what we do. Normally, we look at the steps and we try to find greener solutions with respect to chemistry as well as greener technologies and engineering. If you look then at the new chemistry, which we used then in the second generation, we were able to reduce the overall PMI from 265 kilogram to 156 kilogram, which is about 40% improvement in mass efficiency. And you do see this with the, let me see, somehow. You see it here with the second bars. So the first bars are the original chemistry, and then the second bars you see that we have quite a significant reduction in process mass intensity. And as I said, we don't stop after launch. We looked for further reduction of the PMI to 126, and we were able to do this by looking for possibilities for solvent recovery. Again, solvents really play a very imminent role in the greenness of your process. And as you can see, the average step PMI then dropped to from 26 to 12, and this is really what we are heading to. We call something green if it has a step PMI between 10 and 15, and as you can see, we are now at 126, and we nearly have achieved our goal of having a PMI of less than 100. The next example shows you Januvia. Januvia is on the market for the treatment of diabetes. And this is really, people didn't think that this is possible. We were looking at the last step of the synthesis of Januvia, and I'm only showing you here the last step. In my more technical focused talk on Wednesday, you will see all the background of this reaction. So what we did here is we used the biocatalytic asymmetric synthesis of chyolamines from ketones. And normally your body does this all the time. Alpha keto acids are transformed with transaminases to amino acids. But unfortunately, our ketone, which you see here, and I'll show you in a minute, is not really anything near really the normally natural keto acid. So it's not really, it's not a surprise that with this, what is shown here, the original enzyme, that we do not have any activity. So what you need to do is you have to do evolution of the enzyme. What you do is you change the shape of the enzyme in a very in a manner which allows you to adjust it so that in the end your molecule really fits into the pockets of the enzyme, of the evolved enzyme very well. And in addition, it fits in a way only in one orientation. And then the transformation of the transaminase from the keto function to the amino function can be done only in one orientation, and that means it will be done in a very enantioselective way. So this is the chemistry. So this is the precursor of Januvia. You have the ketone here, and as you can see, it's very different from the natural occurring alpha keto acids. And, but we were able to do this transformation with the evolution of the enzyme, and this has been done by Merck in very close collaboration with Codexis. And as you can see, we got this transformation to work. We are using it commercially on a multi-ton production scale. And it's a lot more productive. It doesn't use any metals because originally we formed an imine here and we did an asymmetric reduction, which has a couple of drawbacks from an environmental standpoint. We are using rhodium, and there is not an abundant use of rhodium. So biocatalysis by itself, by nature, is a greener approach to it. And again, it's lower cost because, as said, the best chemistry has the green aspect, the green engineering aspects as well, and I don't know any examples where it's of higher cost. This is what I wanted to show you from the Merck examples. Now I'm showing you 3 other examples outside the pharmaceutical industry, and I really hope that it will inspire you to think differently, to think out of the box so that we do find solutions and reduce our energy consumption, and we are not going to get more earth, so we really have to make the best use of what we have. Resource productivity innovation. So Lima, Peru lacks of adequate drinking water, but has very high humidity. So there was a local university with declining enrollment, and they installed a billboard to advertise innovative water condensing technology, and it converts moist air into clean drinking water. So it's doable. Think differently. There are solutions out there. Another one is the 3D printing. It makes a 3D solid object by depositing successive layers of filament using a digital model. It uses fewer raw materials. It's constructive versus reductive process. It uses 41 to 64% less energy than making products abroad and shipping it to the US, because this counts as well. The filament is made from renewable resources and can be reconstructed waste-free. It's cheaper, it's more efficient, it's no retail markups or shipping costs. Think differently, innovate. We all can do it. The last example is from Levi Strauss. It's an initiative called Waterless Jeans. It uses 100% recycled water in its fabric dyeing and washing operations. It reduced 770 million liters of water since 2011, and this is an equivalent to supplying drinking water of 811,000 to people for 1 year. I think it pays off. Think differently. The key takeaways from my presentation today is good science, I think, is the key to sustainability, the key to green chemistry, and low-cost manufacturing across the globe. If we focus on developing the best chemistry, then almost always this leads to the lowest cost and the greenest process and a minimal impact on our environment. We think that education and collaboration across industries and innovation are the keys to advancing green chemistry and green engineering. We must think differently in order to meet our future needs. We have one Earth, we don't get more. Within the pharmaceutical industry, I think we are in a very privileged position to help patients do great science and minimize the impact on the planet. In the end, the sustainability of the pharmaceutical industry is dependent on the sustainability of our global population. Well, a few of us are green by nature, but all the others have to and need turning challenges into green opportunities. I thank you for your attention. I'm happy to take your questions.
Okay, we got about 4 minutes for questions, so anybody have any questions for this young lady?
Thank you.
Nick Gallagher from Dublin City University. Just a question at the end about the 3D printer. Surely there's a cost associated with shipping originally the 3D printer so that you can make the object. So isn't the real question, how many objects do you have to make to break even compared to the cost of originally having the resource to perform the task?
That's a very good question, and unfortunately, I cannot really answer that. I'm not really an expert in 3D printing. The only thing I really want to do here is, with this example, to inspire you. But you are absolutely right. You have to look at the whole picture. And this is one thing which is the advantage of doing a life cycle assessment, where you really look at the overall picture. But I'm afraid I cannot answer your question. Okay. But hopefully I inspired you to think out of the box. Oh, sure.
About the processing in decreasing the use of solvent, and I saw the Merck from 256 reduced to 150 something, and then go down to 112. That means your process run in more concentrated condition, or you using some other substitute or some other solvent to substitute your original solvent?
It's a very good question. I think it's a mix of everything. When we look at our processes and making them ready for commercial and being used for launch. And after that, we are looking at solvents because solvent is a main contributor. We are looking at the kind of solvent. We are looking at the number of solvents. So ideally, you don't have more than 3 or 4 for an entire process. We are looking at the possibility to recover solvents. So you have to be very careful which solvents you add into a process. What we also look for is we look at the whole process holistically and see how many solvents do we really need. What is the solvent of the next step? So what we try to do is, once we understand our process, not to isolate as well, but to carry it through. So, and for example, let's say we are using ethyl acetate in the first step, so we see, do we have chemistry in the second step which can use ethyl acetate, and so that we do not have to isolate. Well, in the beginning you have to isolate because you have to understand all the impurity profiles, but once you have this process understanding, you can do things like that. You can run it more concentrated. This is another way of doing it, but you can also, you can look for new chemistry. Maybe you find chemistry using less. So it's really a mix of everything to get this process mass efficiency numbers reduced.
So it's the recovery and the sequential run of your reaction. So that solves the problem. Is that
it's a combination, I would say.
Okay.
It's using new chemistry. It's using looking for the right solvents, looking for the solvents which you can recover, which don't form azeotropes, for example. Using one solvent instead of 3. It's a mix. I would say it's really looking at it from a holistic point of view.
Thank you very much.
Okay.
Yeah, I'm Rakesh from Dr. Reddy's laboratories. I've got a very specific question regarding Januvia. So do you have any idea about the dilution factor at the scale when you do this asymmetric reduction by using enzyme?
Yeah, it starts on a very small scale and doing the evolution, and as said, has been done in Merck, but in very close collaboration with Codexis. So you do this on a very small scale, and then once you see your activity and you do more evolution with the enzyme, and then you slowly scale it up once you see the activity you would like to see.
Yeah, I'm just talking about the dilution, like what concentration this chemistry
I think
is
in the end, I had it on my slide, it's very amazing that you can get to a very high concentration. You start very dilute, but in the end, it's relatively high. It's absolutely usable in the industry, which people thought is not possible to be done, but the enzyme is, the evolution is done in such a way that it is get more stable to organic solvents, and the other way you can do this, you can immobilize these enzymes, and then you can use any solvent you want. You really are not restricted by the aqueous medium. But it's very amazing what you can do with this evolution process.
Thank you.
Okay.
Right. Thank you once again to Dr. Mergelsberg for the presentation.